Today I'm talking about some of the basic concepts in genetics. The first thing is diploid versus haploid cell. A diploid cell is a cell that contains two sets of chromosomes, one from each parent. These are usually somatic cells which are divided by mitosis, or they are produced by the process of mitosis. And haploid cells are those cells that contains one set of chromosomes. They are usually germline cells and they are produced by meiosis. Genes versus allele. Gene is a segment of DNA that contains specific sequence of nucleotide that provide instructions for making proteins or RNA molecule. And alleles are different versions or variation of a gene that arise due to slight changes in the DNA sequence. For example, the DNA here and here, this is gene for eye color, and how their DNA vary between individuals or in a population, that is now allele. Um, While all alleles of a gene occupy the same location on a chromosome, they may differ in their specific nucleotide sequences, leading to different outcomes in phenotype. In other words, we can say that this is one allele and this is another allele of the same gene. When we relate it to Pepsi, for example, Pepsi is a gene, and this different type of Pepsi, these are alleles. In eye color, these are different alleles of a same gene, eye color. Homozygous versus heterozygous. When there are two identical alleles, identical alleles, for example, in this case, this is homozygous, and these identical alleles are at a specific locus. And when there are two different alleles at a specific locus, now this is heterozygous condition. Recessive versus dominant. A recessive allele is an allele whose effect is masked by the presence of a dominant allele. Its associated trait is only expressed when both alleles are recessive. For example, these are two alleles, and when these two alleles are present, then the phenotype is here, and this is now it means that this allele is a recessive allele when both of the when both of the alleles are there, then the phenotype is there. Now look, in this case, that this is one allele, this is another allele, and here this allele is present, but its effect is masked by this allele. So now this is a recessive allele, and even in the heterozygous condition, this allele is expressing, now it is a dominant allele. So a dominant allele is an allele that expresses its effect in the phenotype even if only one copy is present. So in short, in the heterozygous condition, when the allele which is expressed or which is showing its phenotype, that allele is a dominant allele, and in the heterozygous condition, the allele which has no phenotype, or its phenotype has been masked by another allele, that is a recessive allele. So this is the difference between dominant and recessive allele. Autosomes versus sex chromosomes. Autosomes are chromosomes unrelated to sex determination, while sex chromosomes are chromosomes that determine biological sex. In terms of their numbers in human, 22 pairs of autosomes are in human and one pair of sex chromosome. In terms of their inheritance pattern, autosomes equally from both parents, while sex chromosomes, mother always contribute X, father contribute X or Y. Homologous in both sexes, yes, the autosomes are always homologous pairs, and the sex chromosome, no. X and Y are not fully homologous in males, while XX in females are homologous. Trait control, and the autosomes control general body traits and function, while the sex chromosomes control the sex-specific traits and sex-linked characters. Now these are, for example, autosomes and the sex chromosome, XX female, XY male in human population. These are sex chromosomes. So these are only one pair, either this is present or this pair is present, while the autosomes are 22 pairs in human. A law of segregation, which was proposed by Gregor Mendel, it states that each organism has two alleles for a given gene, one inherited from each parent. For example, these are two parents, and these are the alleles, one coming from each parent, and now this is the first filial generation. During gamete formation or meiosis, these two alleles are separated, or they are segregated, mean these two alleles are separated and these two alleles are separated. Only one allele go to the gamete, so that each gamete contains only one allele for each gene. And the alleles recombine randomly during fertilization, restoring the two allele combination in the offspring. And so parents, two alleles for each a given gene, and male, female, and then during gamete formation they become the alleles are separated, they are segregated, and when during the fertilization process they are randomly recombining, restoring the two alleles combination in the offsprings. And when go to the P2 or second parent's generation, then again these alleles for each gene are separated, are segregated, and they go to the gametes and randomly combine. And in second filial generation, the ratio is 1:2:1 genotype, or 1:3 phenotype. So this is law of segregation, which states that, in short, that the alleles are two for one gene, and they segregate during gamete formation and recombine during fertilization, and the recombination is random. Law of independent assortments. Now, law of segregation was about one gene and the number of alleles there, while the law of independent assortment is not about just one gene, rather it is about two or more than two genes, and it states that allele of different genes assert independently of each other, resulting in the 9:3:3:1 ratio in the F2 progeny from a dihybrid cross. For example, this pea plant seeds are yellow and round, and these are wrinkled and green. And their F1 generation is yellow and round, because yellow is dominant over green and round is dominant over wrinkle. And when for the F2 generation the gametes are formed, so 4 gametes are possible from male. A, B, these are the both dominant, simply for yellow and round, and this is for yellow and wrinkle, and this is for green and round, and this is for green and wrinkle. So these 4 gametes are possible in male and 4 gametes are possible in female, and when they are combining in different ratios, so their resulting phenotypic ratios is 9:3:3:1. It means that in the dihybrid cross in the second generation, the 9 will be yellow and round, the 3 will be yellow and wrinkle, the 3 will be green and round, and the 1 will be green and wrinkled. So this is called law of independent assortment, means that different genes assort independently of each other. Type of mutations, here I am talking about single nucleotide polymorphisms. For example, this is the genetic code for in a DNA, and this is a sequence of amino acid which is translated from genetic codes. For example, phenylalanine here, and GCU encodes for valine, and then UGU for cysteine, and AGA for arginine. Now this is the normal sequence of amino acids. When mutation occurs, for example, a single nucleotide polymorphism is happening here, the U is replaced by C. So now when it happens, then the UUC, initially it was UUA in normal human population, and the amino acid is phenylalanine. But now the U is replaced by C, but UUA and UUC both codes both are codes for phenylalanine. So due to this mutation, there will be no change in the sequence of the amino acids. Now this type of mutations are called silent mutation. And for example here, now instead of U, which is in normal gene, now here the U is replaced by adenine. Right now, UUA now encodes for leucine instead of phenylalanine. Now, this is a mutation which is changing only one amino acid instead of phenylalanine, which is in normal population. Due to this mutation, that individual has leucine. So this type of mutations are called messense mutation. And in this case, again, this is the normal sequence, and in this normal sequence, now in this position, this U has been replaced by A. Now when this U is replaced by A, the code is UGA. It is a stop codon. So now in this mutation, the amino acids which are coming after this code, they are not the part of the protein sequence. Now this type of mutations are called nonsense mutation, because a premature stop codon happens and it stops the elongation or the translation of protein amino acids after that code. So this type of mutations are called nonsense mutation. And in this mutation, which is the fourth type of single nucleotide polymorphism, again this is the normal sequence, and here this U has been deleted, and now the deletion of this U results that this G will come here. So instead of now UUU, the new code is UUG, and it will code for leucine. And what will happen now? This is CU, it is here. This nucleotide will come here. So now this is CUU. So valine is now replaced by leucine, and now here This was in normal amino acid, it was UGU. Now a frame is shifting, so this is now GUA. So now it is valine. So in this way, whole of the sequence of amino acids in that protein will change. So now this type of mutation is called frame shift mutation. So in the silent mutation, although there is mutation in the code, but the sequence of amino acid is not changed. In the missense mutation, one nucleotide is changed or replaced by another nucleotide, resulting a change only in one amino acid, which is happening on that code or codon. And in nonsense mutation, a premature stop codon is there, so that the nucleotides which are after that stop codons, they are not being translated. And in the frame shift mutation, when one nucleotide is deleted, then a complete frame shift happens, so that all of the amino acid in the sequence are now different from the normal one. These are some of the example or definitions. The silent mutation, a substitution that does not change the amino acid sequence, which is happening here. Missense, a substitution that changes one amino acid in the protein, which actually happen in case of sickle cell anemia. And nonsense mutation, a premature codon or a stop codon is there, just like Duchenne muscular dystrophy. And frame shift mutation alters the entire reading frame, often causing a non-functional protein. Tessage disease is an example. Okay, autosomal recessive disease. An autosomal recessive disease is a genetic condition that occurs when an individual inherits two copies of a defective gene, one from each parent. These conditions are caused by mutations in genes located on one of the 22 pairs of chromosomes, which are autosomes. People with one defective gene, a single mutation, are called carrier, and they usually do not exhibit symptoms of their disease. For example, there is a pathogenic variants mutation here, but it is just a carrier. The person is not suffering from the disease. Again, this parent, again, it has a mutation, but the disease is not there. So now this is autosomal recessive disease, which is only happening when the two of the deleterious or two of the harmful mutations are there, while only one copy of that gene results a carrier individual, and which is phenotypically not affected. So this type of disease is called autosomal recessive disease. While in case of autosomal dominant disease, only one copy of the allele or one mutated copy of a gene is sufficient to drive the phenotype. So this parent is affected, this gamete, the child which is coming from this gamete will be affected. So if any autosomal disease, when it is manifested, when it is phenotype is there, in the presence of only one copy of gene, that is called autosomal dominant disease. And when one copy is not bringing up the phenotype or the disease, and the disease is happening when both of the copy of gene are there, such disease is called autosomal recessive disease. Penetrance versus expressivity. This is another concept in genetics. Penetrance is the percentage of animals of a specific genotype who express the phenotype associated with that underlying genotype. Now, for example, this is the genotype to allele of a gene, and these are the trend of that genotype in a population, or now it is called penetrance. For example, there is a there are 100 people, and out of 100 people, 2 of them have a genetic disease. So that mutated gene is now 2% penetrant in that population, while expressivity refers to the degree that a particular genotype is expressed as phenotype within an individual. For example, this is a deleterious mutation and it is bringing a phenotype. Now, how severe that phenotype is, that is the expressivity. So penetrance is a phenomenon related to population, and expressivity is the phenomenon related to an individual. And penetrance in terms of the number of the the percentage of animals of a specific genotype who express the phenotype, while expressivity is the degree that a particular genotype is expressed. Thank you so much. These were some of the concepts, basic